Hello. In today's class, we'll talk about the structure of the DNA. So, structure of the DNA. Yes, this topic will be precise. It will help students to understand the basic structure of the DNA that they need for O level as well as S level. So let's get in. DNA is actually the chemical. The DNA is chemical that is making the chromosome. So what types of chemical is this? Actually, DNA is composed of three major things. One is nitrogen base, other is sugar, a pentose sugar, you can call them deoxyribose sugar, and phosphate. Okay, those three things are the basic elements of the DNA. And yes, when it is formed, when it is arranged, some specific bonds are formed. We'll talk about that. So let's get in the structure of the DNA. So we've just learned there are nitrogen base. Actually, there are 5 nitrogen bases, which is represented as A, T, G, and C, along with U. I'm writing U apart from these 4 because U is not normally found in the DNA. U, that means the uracil, it is for the RNA. As we are discussing the structure of the DNA today, so actually it is about A, T, G, and C. So A means adenine, T means thymine, G means guanine, and C means cytosine. Yes, out of these bases, okay, some are called purine and some are called pyrimidine. So that part will be discussed in the class. So you see there are 4 nitrogen bases for the DNA. Now how it is made, let's get to the point. So one nitrogen base, think of it, I'm writing nitrogen base as N actually. Think of it, one nitrogen base, one sugar, and one phosphate. All together they make a structure and we call them nucleotide. So you see, nitrogen base, sugar, and phosphate, they are making nitrogen base, sugar, and phosphate, they are making a structure, the nucleotide. So nucleotide is also called the basic unit of DNA. Okay, but DNA is made of many nucleotides, so DNA is also called the polynucleotide strand. So how it is formed here? So think of it, I'm just adding more. Think of it, another nitrogen base, sugar, and phosphate. So this is another nucleotide. Nitrogen base, sugar, phosphate. Another nucleotide. Nitrogen base, sugar, phosphate. Yeah, you can go on actually. So I'm just writing this 4 of this part as there are 4 nucleotides, so that I can show 4 nucleotides, 4 nitrogen bases here. But you can carry on. DNA is a very long one containing hundred thousands of nucleotides. Okay, so you see, this is nucleotide, nucleotide. So what really happened, one nucleotide is actually joined with another nucleotide making phosphodiester bond. So what really happened, you see, if I use the different color, the sugar of one nucleotide and the phosphate of another nucleotide make a bond. You see the red line, this is the phosphodiester bond. Phosphodiester bond. Okay, so the same thing happened, the sugar and phosphate making the one phosphodiester bond, sugar and phosphate making the bond, that is the phosphodiester bond. And this is how the student a strand of the DNA is made. Normally, when you see the DNA, you see this types of structure, isn't that? So you see, this is called a strand. Strand number one, strand number two. So DNA is made of strand. So this strand is actually this one. It is made of many nucleotide joined by phosphodiester bond. Okay, and now if I make it pretty simple, removing some excess part, it will be looking a bit precise. So you see, this is how one of the strand of the DNA is made. But you all know the DNA is a double-stranded things, so I should draw another strand. If I rub these things to get some space, so that you can see the whole structure in a single piece. Okay, so I'm writing another strand. Suppose nitrogen base, sugar, and phosphate. Okay, nitrogen base, sugar, phosphate, and so on. Okay, and very similarly, the bond is formed, the phosphodiester bond. I'm using the same color to represent the phosphodiester bond. You know that sugar and phosphate, a phosphodiester bond, sugar, phosphate, and so on. Actually, it will be continued. So you see, the two strand, I mean the DNA that you normally see in that way. Okay, strand number one, strand number two, or vice versa. So think of it, this is strand one strand, this is another strand. How this strand is made of, you already know this part. But this is not the end of the story. When two complementary strands are arranged parallelly, what really happens? A bond is formed between nitrogen bases. You know, the nitrogen bases of one strand and nitrogen base of another strand. You see? So this is what nucleotide. So the nitrogen base of one nucleotide of one strand is making bond with the nitrogen base of another nucleotide of another strand, you know. So it is making bonds. You see, I'm using the black color to represent the bond, and this bond is called hydrogen bond. You see, that bond is called hydrogen bond. Don't forget, so this bond that was connecting or attaching two free nucleotide was the phosphodiester bond, isn't that? Phosphodiester bond. Yeah, in this is how hydrogen bond is formed, like that, like this, like that. Isn't that? Okay. And you already know there are 4 nitrogen bases for the DNA. So actually there is nothing like N. We used N here as a representative of nitrogen bases, but nitrogen base exists in 4 forms for DNA, like A, T, G, and C. So I should not write N, I should write any of it replacing the N. Is that? Okay, so I'm just doing the same thing. Think of it, there is no N over there. There should be nitrogen bases. Example of nitrogen bases. I'm giving the common, leaving the common word. Common, you see. For example, just imagine one of the nitrogen bases is A. So yeah, the another one is T. This is random one, obviously, in your DNA, in your gene, it would be arranged specifically. I'm just giving here as a structure. So C. So you see. So if one of the nitrogen bases is A of one nucleotide, as it is binding with the nitrogen bases of another nucleotide or the other strand, so that means it is following the base pairing rules. You can tell me, sir, what is base pairing rules? You know, base pairing rules. That means it has to be T over there if it is bonding. A always bind with the nitrogen base T. T, it has to be A. If it is G, it is C. If it is C, it is G. And yes, when A and T is binding, 2 hydrogen bond is formed. And T and A is binding is same. G and C is binding, 3 hydrogen bond is formed. And this is how you get the structure of the DNA. So I'm sure it is pretty easier. I try to make it concise and short so that you don't get bored. Yes, if you repeat these things again, you see, sometimes when you see the DNA structure in the book in a simple way, you see this types of structure. kind of. ATGC. Yeah, it's a random one. I'm just taking it, like that. So you can tell me, sir, how this complicated or more structured structure is related to this one. Yeah, if I try to make it simple, you see, if you look closely, wherever you go, the phosphate sugar is pretty same. The phosphate and sugar is same. The phosphate and sugar is same. Not only in your body, not only the human body, in any living organism, this structure is same. Is the phosphate sugar, phosphate sugar. So you see, this is pretty same. Every cases. Only the thing that is varies the nitrogen bases and their arrangement. So when you draw the DNA simply, you don't care about this structure. So we simply actually give a line for that. Okay, for example, we give a line for that. Give a line for that. Only we keep the nitrogen bases that is unique. So that's the reason you see these types of structure. So you see, actually, this one is the sugar and phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay, so only these varying things are presented. Okay, the student. Thank you very much.